Okay, thank you, Laura, for the introduction. It's really, really a great pleasure that I can speak at this seminar series. So, yeah, I mean, as requested by Junior and also Laura, I changed my talk a little bit to also include my work on RNA structure, RNA protein interactions, and also some of my recent research on protein structures, but all of that are wrapped up in with the name of big data analysis with machine learning. Okay, so that's my talk. Next generation structural biology powered by big data and machine intelligence. Okay. Yeah, just very briefly to introduce myself, because I think this is a great time to meet all of you. So as you can see from my screen, all I want to highlight is that I originally started computer science for my first PhD, and I got the degree from University of Science and Technology of China. And then I moved to Colombia to study biology from the beginning. So I got another PhD degree in biophysics for 6 years. And I spent a few months in Colombia for a very brief postdoc to finish some of my graduate research. Then I moved to Stanford with my postdoc research and working with Howard Chang. When I was in Colombia, I mainly studied computational structural biology, and I worked with Barry Honig. And as I just chatted with Julian, and maybe we had run into each other when we were both in Colombia. Yeah. And when I was in Stanford, I changed my research area from computational structural biology to more focused on genomics, but still using computation as my main tool. Then just 6 years ago, I came back from United States to join Tsinghua University as an assistant professor, and was promoted to associated professor 3 years after that. And I'm now running a hybrid lab in Tsinghua University. I do have people working with me, mainly focusing on computational side, and I also have people working with me focusing on the bench. So I'm running a hybrid lab. So that's a very brief introduction for me. And still, I'm mainly focusing on structural biology, but to me, structural biology is not, well, Cryo-EM, not a crystallography, but I'm using computation and also sequencing to approach this study. Okay. Yeah. So I worked in chromatin structure and also RNA structure and also protein structure. And for chromatin structure and RNA structure, we mainly using sequencing to obtain the data. And for protein structure, I'm working on Cryo-EM data analysis. Okay, so I include 3 small stories that cover that cover all 3 aspects of the slide. Just this one, okay. So my first piece of work is about RNA-protein interaction prediction using RNA structure data and machine intelligence. So RNA-protein interactions are at the center of RNA functions and regulation. So indeed, from transcription to processing to translocation, and also translation and degradation, the whole life cycle of RNA molecules is dependent on their interactions with their protein partners. So identification and understanding protein RNA interactions are the key to study RNA functions. Actually, the top one important technology to resolve the complex network of cellular RNA protein interactions is CLIP. CLIP has many technology variants, but the basic principles are about the same, shown here in a very nice review. CLIP is powerful and has led to many important discoveries on RNA regulation and functions. But we also have the experience that it is a very difficult experiment to perform. So it is labor intensive, it's expensive, and also have some very strict requirements on, for example, on antibody and also on sample qualities. An even more annoying reality for studying RNA protein interactions using CLIP is the knowledge of RNA protein interactions obtained in one condition cannot be directly transferred to another condition. So in other words, RNA protein interactions are really heavily depend on the cellular conditions. And as we have examined the RNA protein interaction data for the same protein, but obtained in different cell lines, we found the overlap is very low, as you can see here, between the HepG2 and K562 cells. For most of the proteins, only just a very small fraction of RNA protein interactions are shared, are common in the two types of cells. So the majority of the interactions are actually tissue specific, as you can see, shown in red and also blue. So this tissue specificity also explains the limitation of using sequence motifs to capture, or in our computer science vocabulary, to model RNA protein interactions. This sequence motif idea actually is borrowed from the studies of transcription factor bindings on DNA. But we found that use this degenerative sequence motif to model RNA protein interactions is very crude. So most of the matched sites are actually false binding sites, as shown here in blue. And it also misses many true real binding sites. The reasons are many. Okay, but a key point is sequence alone cannot determine the binding of a protein onto its RNA targets. In addition to sequence, most proteins bind to RNA with its preferred structure. Some tend to bind, for example, a single-stranded region. Others prefer to double-stranded region. Some may favor stem loops. The others may like bulges. And RNA structures, however, are quite dynamic in different cellular conditions. RNA structure changes affect protein binding and is a key factor for the tissue-specific RNA protein interactions. So we already have some ideas on how RNA structure determine RNA protein interactions, mainly coming from tertiary structure studies. But the principles in general, and especially in cells, is unknown. So the major hurdle is a lack of efficient methods to obtain RNA structure information, especially within cells. Several years ago, when I was in Harvard lab, we co-invented the IcSHAPE technology, which uses a small molecule shown here. The name of the molecule is NAI. The molecule can specifically modify a single-stranded region of an RNA molecule within cells. Very nicely, this modification can stop or interfere RNA reverse transcription, thus can be read out by sequencing and bioinformatic analysis. Okay, so as you can see, the modified site actually corresponds to the single-stranded region, and all this information are determined by sequencing and bioinformatics analysis. So I will not explain the detail of this technology. Just very recently, we had extended the technology to use reverse transcription mutation for signal reading. This improvement is very important for studying structures of small RNAs, including RNA fragments and functional sites. So this IcSHAPE and also IcSHAPE mag technologies are very powerful in that they can be used to obtain accurate information of transcriptome-wide RNA structure in cells for all 4 nucleotides. In the recent study in my lab, we used IcSHAPE to study how RNA changes its structure during its life cycle within different subcellular locations. For example, chromatin-associated nuclear plasma and cytoplasma. By comparative and association studies, we quantified the structure changes and established a central role for RNA structure in connecting different RNA events, including transcription, translation, and also degradation. In another study, we used IcSHAPE to obtain RNA structural maps from zebrafish eggs and early embryos. Again, by comparative studies, we observed hard regions of RNA structure changes, and then we constructed a structure-based RNA regulatory network. We identified and validated protein regulators on maternal RNA degradation by analyzing this network. So we were interested in the interplay of RNA structure and RNA-protein interactions. We resolved RNA structure in 7 cell lines and observed substantial structure changes, as you can see, in different cell lines. Interestingly, when we align the RNA structure profiles along with RBP binding profiles obtained from CLIP studies, we observed a strong correlation between the 2 changes. In other words, different RBP binding sites in 2 cell lines likely have a different RNA structure in the 2 cell lines. This observation is not a surprise to me and likely to many of you, given that we know RNA structure affects RBP binding and vice versa. But anyway, this is the first time we observed the detailed landscape of this association, and actually for many different proteins and on a transcriptome-wide scale. So shown on the right of this slide is an example. It shows that how RNPM binds differently in the 2 cell lines, HAPT2 and K562. This differential binding is affected by the different RNA structures at the binding sites. Okay, so this observation inspired us that we maybe can use RNA structural information to predict RNA-protein interaction changes. So we envision that we can construct an interaction model using CLIP data and RNA structural data obtained. For example, in condition 1, okay, so in condition 1, we have both the CLIP data and also the RNA structural data. Then we construct an interaction model. Then we can extend the model to condition 2, okay, here, to predict the interaction changes using the RNA structure measured in condition 2. Okay, so you see the structure is different, and then the binding is also different. So this is very nice because once we have performed CLIP experiments in condition 1, we can predict RNA-protein interactions in all other conditions where RNA structures can be measured, okay? And once we measured RNA structures in the new condition, we can readily predict interactions using RNA-protein interaction models that we have built. This is powerful because in the new condition, we only have to measure RNA structure for once, but we have hundreds of RNA-protein interaction models built in other conditions, we can leverage those models to predict RNA-protein interactions in this new condition. So essentially, we can obtain hundreds of RNA-protein binding profiles in this new condition. Okay, but this also, but I mean, it must be an accurate prediction, otherwise it is meaningless. So now the question is how to achieve an accurate model that combines RNA-protein interaction information and RNA structure information together. So we turned to deep learning, a most powerful AI technology that appeared in recent years and quickly changed many areas in science and technology, and even including our life and even recreations like here in the Go game, okay? So for most cases, the power of deep learning is a philosophy of using complex models that in principle can model all possible mathematical functions. And the success of most complex deep learning models relies on using big data to train the model. Indeed, without big data training, okay, the complex model often performs even worse than traditional machine learning methods, which now sometimes referred as shallow learnings. Luckily, when we have a huge amount of data, we can construct a deep learning model that performs much superior to shallow machine learnings. And very fortunately, we do have a huge amount of data that we have measured for RNA-protein interactions and also RNA structures using CLIP and IcSHAPE. Okay, so we built upon the cutting-edge deep learning technologies and constructed a complex interaction model trained on RNA-protein interactions and also RNA structures in the same condition. Okay, this I have emphasized this for quite a few times. We need the data in the same condition to construct the model. Finally, for any protein, once we have trained a model and also have measured RNA structure in the new condition, we can use the model and also the RNA structure in the new condition as the input to predict RNA-protein interactions in this new condition. Okay, so this is a key idea. So we called our deep learning tool PrismNet, which stands for Protein RNA Interaction by Structural Modeling using Deep Neural Network. So we show that PrismNet can accurately predict RNA-protein interactions. This slide shows one example protein, that is IGF-2BP1. We built a IGF-2BP1 RNA interaction model using eCLIP data from Jing Yao's lab and also RNA structure data from our own lab. Both are in K562 cells. Then we applied this model to predict IGF-2BP1 binding in HepG2 cells using RNA structural data in HepG2 cells. We compiled our prediction with eCLIP data also obtained from Jing Yao's lab in HepG2 cells as positive references. Okay, as you can see, for IGF-2BP1 binding on transcript EIF3F, PrismNet predicted all 10 binding sites. We compared our predictions with other tools, DeepBind, GraphProt, RCK, and we observed that the other methods predicted fewer true binding sites with some false positive predictions. Okay, so this is just one example. Actually, we have carried out the analysis for almost about 200 RNA binding proteins with available CLIP data. So we found PrismNet consistently better than the other prediction methods shown here. So one extra, so the most important point that need to highlight is that we found only experimentally measured RNA structure improves prediction. So indeed, the above mentioned methods did use predicted structure in their predictions, including graph plot and also RCK. So to investigate further, in addition to the real PrismNet, we also implemented a few other variant predictors. So with only sequence and also with only structure, and both sequence and predicted structure, not the real structure, but a predicted structure, and also sequence and random structure as the input. So we found that the real PrismNet performs the best, and the structure only PrismNet performs the least. But very interestingly, PrismNet using PrismNet variants using sequence only or predicted structure or random structure, they all perform very similarly. All of them are worse than the real PrismNet. So our explanation is that the predicted structure is somewhat second-hand information that is already buried in sequence. So if your model has been very good, the usage of additional information of predicted structure adds no more value onto the sequence input. So that's our explanation. Okay. So on the right, you can see the PrismNet variant using only sequence is always worse than the real PrismNet without the experimentally narrowed structure. The design of PrismNet allows us to employ deep learning attention analysis to obtain positions that are most responsive to a predicted interaction, so which actually indicate accurate protein binding site for this interaction. So this slide shows some examples from the protein RB-FOX2. As you can see, there are some hard regions on the predicted binding sites, some hard regions. Okay, so these hard regions are actually correspond very well to the real binding site with a similar sequence motif. Okay, but we have colored the responsive locations in using different colors, so mainly showing in red. Okay, so we see that both sequence and also structure are responsive in these examples. Okay. We set out to validate that both specific sequence and specific structure are important to protein RNA interaction. The example we chose here is TIA1 binding, which tends to bind single-stranded polyUs. As we know, PrismNet predicted the binding of TIA1 to a transcript region with a score 0.9. We validated the interaction, see on the figures on the right. When we mutated the sequence at the responsive site, okay, we see a dramatic decrease of the predicted score to 0.06. Again, we validated the much decreased interaction. And we also mutated the sequence at a distal place, which does not change the binding score too much. And we also validated the interaction. So all the above investigations were done in keeping the site in a single-stranded conformation. So finally, we slowly cooled down the construct and actually the distal mutation to let it fold into a double-stranded conformation. Okay, so we see that although the foci sequence is not changed, it's still poly-U, the interaction now is much largely disrupted as you can see from our prediction, and also as a which is the consequence of the change of the foci structure. Okay, it's also validated here. Okay. Actually, we have done more experiments to see how RNA structure changes affect RBP binding. So here we used SND1, a RBP that usually binds to double-stranded RNA. We see that with the same sequence, actually they are same sequence, but we managed to obtain different structure by using quick cooling and also slow cooling, we can obtain different conformations of the structure. And we see that in different structural conformation, the binding are different. So which very nicely validated that the structural conformation is very important for RNA-protein interaction. Okay, so finally, in total, we made RNA-protein interaction models for about 200 proteins, okay, obtained by using the clip data obtained in different conditions. And also we measured the RNA structure in the matched conditions, okay, actually in 6 different cell lines. So by the above mentioned attention analysis, we retrieved the sequence and the structural integrative motif for all these 200 proteins. So we organized them into a big compendium based on their similarities shown here. These integrative motifs are degenerative models derived from the complex PrisMNet models. However, they are very straightforward in describing how a protein prefers what kind of RNA sequence and structures to bind. So a direct usage of this integrative motif is to use it to search for possible binding sites on the transcriptome when RNA structure information is available. So indeed, we see that integrative motifs are a few times more accurate than traditional sequence motifs in identifying a true binding site. As you can see here, we observed a few times more higher accuracy. But by the way, we found that the sequence component of the integrative motifs, so the sequence component, the structural component, this sequence component are very similar to the known sequence motifs from other studies, or they are obtained quite very differently. So we obtained the integrative motif from a attention analysis from our deep learning models. Okay, so this is very good. Okay, so in the next piece of the story, I want to demonstrate the usage of PrisMNet in predicting host factors that bind to the RNA genome of the RNA virus, SARS coronavirus 2. Okay, so simply SARS-2. The whole world has been totally changed by this tiny virus, SARS-2, with a very small genome that encodes a handful of proteins. The whole life cycle of this virus heavily depends on its interplay with the host, especially the interaction between the viral RNA and the host RNA binding proteins. So we set out to study RNA structure of SARS-2 in infected human cells. Using IcSHAPE, we obtain a high quality structural profiles that covers the vast majority of the genome. So here, the data. We constructed a full secondary structural model for SARS-2 RNA genome based on the RNA structural profile. This model contains a lot of information for the virus in cells. For example, we identified many conserved structural elements that are important for viral replication. Actually, we started this in our paper, but this is not the focus of my talk today. Today, I will be focusing on using PrisMNet to predict possible host factors for SARS-2. And indeed, using the 200 PrisMNet models, we predicted many proteins that may bind to SARS-2 RNA, focusing on the UTR, and the number is 42. We validated many of the bindings through comparisons with our own proton mass spectrometry study, and also recently some recent publications, okay, large scale studies. We found very significant overlaps between our predictions and all these experimental studies. We also performed proton Westons to validate individual predictions. Actually, we validated 11 interactions, for the 11 interactions, we validated 9 of them positive. Okay. So the validation rate is very high. And very interestingly, as our predictions are based on both sequence and structure of the RNA targets, so we can validate the RNA structure is essential for the interaction by structural mutation, shown here on the right corner of this slide. Actually, we used a tour virus that we can make our own sequence changes that disrupt the structure. We see that when the structure is disrupted, the interaction between the RNA and the protein is largely disrupted. But when we restore the RNA structure, we see that the interaction is also restored. Okay, so we performed knockdown assay and found that the perturbation of many predicted binding proteins affect SARS-2 infectivity in cells. For example, knockdown of IGF-2BP1 leads to a lower SARS-2 infection rate. So these proteins are functional host factors for SARS-2. There have been many system-wide screenings to search for functional host factors for SARS-2, but our focused screening is very efficient. Moreover, our prediction also generated detailed model for the interaction. We not only have the host factor, but we know where the host factors bind to the viral genome, okay, right? So we designed antisense oligos to target the binding sites and to perturb the interaction. Very interestingly, we found that this antisense oligo also affect viral infection. So that's our prediction, not only identifies functional host factors, it also provides some testable structural models for following studies. Okay, so we just showed the knockdown of the host factors or interfering host factor viral RNA interactions affect SARS-2 infection in cells. All of this could be developed maybe into possible COVID-19 treatment solutions. But however, a more readily antiviral solution is drug repurposing. So we thus searched for available drugs that target the predicted host factors. Okay, we identified quite a few, and then we tested whether they can be used to inhibit SARS-2 infection in cells. We use remdesivir as positive control and DMSO vehicle as negative control. We identify a few small molecule drugs that can significantly inhibit viral infection. So for example, here, okay. We also tested drug toxicity by cell variability. Finally, we found a few drugs that do inhibit SARS-2 infectivity in cells with little, so for example here, with little toxicity. Okay, so we determined more parameters, including IC50 and also CC50 for these drugs. Also, of course, much more work remains to be done for any possible clinical usage of these drugs. But here we only want to showcase that through an integrative approach that combines RNA structural studies and AI-based predictions, we may identify vulnerable antiviral drug targets for SARS-2. Okay, let's get back to CryoNet. We have constructed a website to provide our predictions in 7 cell lines, and also the predictions of RNA-protein interactions already resolved in 7 cell lines. We also provide the 200 CryoNet models for your own usage. For example, if you do have RNA structural data in the new cell line or a new condition, you can use our CryoNet model to make your own predictions to obtain actually maybe 200 different RNA protein binding profiles in your new condition. Okay, so the website is still preliminary, but we are still working on improving the interface and the document. So, but if you have interest in using it or you have any questions, you can just drop me a line. I'm more than happy to help. Okay, so
if
this is the first part of my talk. Yeah.
Yeah, just mind the time. You have about 15 minutes if you want to still have a discussion.
Okay, yeah, so maybe I can just go very quickly on the second and the third pieces of my work. Okay, so as I mentioned, I not only studied RNA structure, but also chromatin structure and also protein structure. Okay, so chromatin structure is one of the bases of epigenetic regulation, which is very, very important for the regulation of gene expression. As you already know, that ATAC-seq has already been invented to very effectively measure the chromatin structure on a genome-wide scale. The technology was developed in my previous postdoc lab by Howard Chang and Greenleaf. Very convenient technology. And just a few years later, after the development of ATAC-seq, they developed a single cell version of ATAC-seq that can deconvolve the chromatin accessibility at a single cell level. Okay. All seems very nice, but many new technologies suffer some new problem here, shown as the data quality. And as you can see, and this is our own analysis, we showed that the single cell ATAC-seq data are very, very sparse, which means a lot of very high level of data loss. Okay, so only less than 10% of the data could be readily measured in most of the cases for single cell ATAC-seq experiments. So how to deal with this, how to enhance the data quality? I mean, we thought about a deep learning framework, the variational autoencoder that can learn the representation of single cell ATAC-seq data in the low dimensional space. Okay, so we developed this tool. We call it the SCALE. It's for single cell ATAC-seq analysis via a latent feature extraction. We combined the variational autoencoder together with the Gaussian mixture model, and we input the matrix of single cell ATAC-seq data. Every row is a peak and every column is a cell, and we try to reconstruct the data using this autoencoder framework. And in the load, I mean, the autoencoder first to reduce your dimension, and then to increase it to reconstruct the single cell ATAC-seq profiles. Okay. So we tested with 6 different data sets. Okay, I don't want to go to the details of the data sets and shown here. As you can see, without the dimension reduction for this single cell data, the data is evenly distributed. The distribution is, I mean, because of the high level of data loss. Okay, but when we applied our scale framework to constrain the data, we have very good embedding for the single cell ATAC-seq data in the low dimensional space, as witnessed by the very good clustering of the different cell types within this low dimensional cell embeddings. Okay. Okay, so for more data sets. So it shows that our scale tool is superior to another deep learning tool, SCVI, on the single cell ATAC-seq data. Okay, so then we can perform clustering on this low dimensional space. And we found the scale performs much better than different tools. Some are developed for single-cell data, attack-seq data analysis, but others are mainly for single-cell RNA-seq data analysis. Okay, at the time we developed the scale, there are no many other tool dedicated for single-cell attack-seq data analysis. Okay, again, tested on many different data sets. As you can see from the confusion matrix, our tool is much better than the other tools. Okay, so also this framework gave Scale the possibility to reconstruct the original expression, original single-cell RNA, attack-seq profiles, as you can see. And this is kind of, in the single-cell data analysis, we call it imputation. As you can see, we have a very good performance on imputation, the data loss. Okay, we can reconstruct the data from our data representation learning using Scale. Okay. The third feature for our framework is our ability to deal with the batch effect in single-cell data. So we actually improved this framework by some redesign of the framework. We developed the SCALEX, and now we can use this SCALEX to perform data integration and also batch removal for single-cell RNA-seq data and also single-cell attack-seq data. So we have tested in many different data sets and also compiled with different very popular single-cell RNA-seq data analysis tool. For example, CRET, Harmony, MN, and Scanorama. Okay, so a very unique feature of our tool is that SCALEX is a global method that tries to learn the data distribution of the single-cell RNA-seq data. So it has an advantage to deal with partial overlap, as shown here. For CRET and Harmony, they kind of messed up with those partial overlap data sets, but SCALEX is not dependent on local recognition of similar cells. It actually learns the data structure from the single-cell data set, and then it doesn't suffer from this mess up from the partially overlapped data sets. Okay, so we actually constructed a simulated data set to confirm these findings. Okay, so with more slides to show that our framework is an online method. I mean, have very good computational performance. It's very computationally efficient, can be scaled to very big data sets. And very nicely, it constructed a universal, global, or even global cell embedding space that can project or newly incoming cells into that cell embedding space. Then we can construct we call it a elastic single-cell atlas. So demonstrated here, showing using the mouse atlas and also COVID-19 data sets. Okay, I don't want to go into the detail, but I can tell you that our integration is aligns very well with some very large scale COVID-19 studies, and we revealed very similar phenomena for the immunoresponse in COVID-19 patients. Okay, that's a second part of my talk. In the last just a few minutes, I will quickly go through my final piece of the study in protein structures. Okay, so we all know that Cryo-EM is a revolutionary technology that can determine protein structure very efficiently for very big protein complexes. But we know that computation is very important for Cryo-EM structural determination by single particle analysis. There have been a lot of tools developed for this purpose, but there has been no good tool developed for the construction of atomic structural models from density maps. Okay, so what's the problem of model building from density maps? So shown here, I'm using a cartoon. So here shows the density maps obtained from Cryo-EMs. So this is not readily readable for all of you, right? Because you don't recognize the densities. We need to fit amino acid or atoms into these density maps. So shown here, you see, I mean, if you can fit the residues into these densities, that's a procedure of model building, okay? So we try to do this in an automatic way. We borrowed a lot of very cutting edge technology from deep learning. So first of all, we try to recognize every single amino acid using some technology very similar to facial recognition. That's in computer graphics, okay, by deep learning. We also employed a visual language method to residues into a chain, okay? So that's a protein chain that finally to construct the atomic level structural models. We even borrowed the some idea of AlphaFold and AlphaFold 2 to incorporate the protein structure prediction for the improvement of our structural modeling pipeline. Okay, so finally, this is our whole neural network. It's a very complex one. It includes the amino acid recognition and also amino acid tracing and also sequencing feature learning. But we put all this together into an end-to-end learning framework. So you input a density map, you will output a structural model. The whole procedure is done by one single deep neural network. So this is very important for the success of this network. So finally, we have come up with this network. We call it CryoNet, that can automatically generate atomic level protein structural models from density maps shown here. It's very, very similar to the ground truth. Okay, as you can see, very small mistakes and very good estimation for the secondary structural elements. And also we can even build up the side chains, which is very powerful. Okay, so we can even recognize some new proteins. So shown here with the collaboration with Yigong Shi's lab, they are experts in the structural studies of spliceosome. So we collab. So in their density maps, there are few proteins that don't know what are they. They don't know what are they. But for our computational pipeline, we can construct the protein sequence and also structures, and we can identify all these proteins they don't know before the collaboration with us. Okay. Okay, with all of that, I would like to thank some very talented students in my group and also my collaborators, because I have used a lot of time. So finally, I want to save a few minutes to interact with you for any questions. Okay, thank you so much.